How's it going, folks? Can you guys hear me okay? All right, welcome back. Happy Wednesday. It is 12:01. We're going to get started, folks. Does anybody have any questions, any issues, any problems, anything at all that you'd like to discuss before we get started? We're doing okay so far? All right, folks. How many of you have started to study for the first lab exam? Okay, see a few hands there. Guys, I can't say it enough. I said it in the beginning of the semester. The first exam is the most difficult exam of the semester. Okay? Because there's a lot of bones and a lot of muscles in our body. So there's a lot of memorization. There is no two ways about it. You have to memorize the name of those bones, the bone markings. You have to memorize the name of the muscles. And if you don't start doing that right now, you will be in trouble when the first exam comes. And the first exam is going to be coming up pretty soon. Okay, we're already on the fourth week of class. Okay, we all know that we have a study guide for the exams. If you go to Lamaku and you can find there under the important resources the exam list for each one of the exam list for the exam one, exam list for the exam two, and the exam list for the exam three. And whatever is included in that list, this is what you're responsible for. If it is not in the list, you don't need to worry about it. It's not going to be on the exam. Okay? But you need to get going. If you start to study the week before the exam, you will fail that first exam. I've been teaching this class for 8 years now. And if there's one thing that I know is the fact that if you're not prepared, you're not going to do well in the first exam. Okay? So please, please, please get started. All right. All right, guys. So we should have a easy lab today. We're only covering one thing, only lab three. As a reminder, for next week, Wednesday, we're going to be covering two labs, 4 and 5, which means that you guys need to turn in two pre-lab assignments before next week, Wednesday. Okay. If you have any questions about the schedule, please look at the syllabus. There's also a course schedule that is attached to Lamaku. You can find that and know exactly what we're going to be covering for each week of class. So let me share my screen with you. And we can go over lab 3. And for the folks that are in my lecture, we have already covered this. We're going to be talking about histology, cells, and tissues, focusing mostly on the cells. And again, if you are taking my lecture together with the lab, this is something we have talked about before, and it's going to be just a review for you, which is not a bad thing since we have a lecture exam. Our first lecture exam is going to be tomorrow. Okay. Now, what we're going to be covering in this lab, we're going to be talking about the different parts of the cell. And I'm going to go very quickly, very briefly, and discuss the different structures of the cell that are found on the outside, as well as on the inside of the cell. I'm going to spend a little bit of time talking about the cell cycle, focusing on cell division, mitosis. What are the different steps of mitosis? What are happening in each one of them? And then I want to talk a little bit about cell membrane and how to move things in and out of the cell. Okay, now in order to understand how to move things in and out of the cell, we need to understand what a cell membrane is made of. So those two things come together. And then we're going to go over our lab activity for today, which is something slightly different from what we have done so far. So today we're not going to watch videos on that puzzle. Today we're actually going to do experiments on a website called the PhysioEX. I'm going to go over with you. I'm going to tell you exactly what you need to do. Okay? All right, we all know that we can divide a cell into three main parts. We have the skin of the cell that we call the plasma membrane. And this plasma membrane is very important, right? Because it's going to separate the inside of the cell, the internal environment, from the external environment, whatever is outside of the cell. Then in the cell, the space within the cell, this is what we call the cytoplasm. And the cytoplasm is made of two things. It is made of a fluid inside of the cell called the cytosol, and this fluid is mostly water. And then embedded in this fluid, swimming around in this fluid, we're going to find what we call the organelles, which are little organs that perform different functions within the cell. We're going to go over a few important organelles. And then finally, we have the nucleus of the cell. And the nucleus of the cell is what we call the control center of the cell. Why? Well, that is because our genetic material, our DNA, is found inside the nucleus of the cell. And our DNA makes who we are. You know, it controls everything in our body. Okay. Now, here we have an idealized little cell. You guys need to understand that the cell here does not exist, okay? But if you could imagine a cell that contains all the different structures, all the different organelles at the same time, this is what the cell would look like. Now, let's start from the outside of the cell and then move into the cell and identify some of the structures and the organelles and what they do. On the outside of the cell, we have this big whip-like structure right here that we call the flagellum. Flagellum is singular, flagella is plural. And we all know that the only cell in the human body that contains a flagellum is the sperm cell. Now, what is the function of the sperm cell, of the flagellum in the sperm cell? The function of the flagellum is to allow the sperm to move, to find the secondary oocyte and fertilize that egg, okay? Another structure on the surface of the cell is what we call the cilium. Cilium is singular, cilia is plural. And the cell that has cilia is going to find cilia all over the surface of the cell. There are thousands of cilia covering the cell. Okay, it's not a single cilium. And both the flagellum and the cilium are made of the same thing. What are they made of? They are made of a cytoskeleton called microtubule. Okay, but they do have different functions. What is the function of the cilia, folks? The function of the cilia is to expel things out of our body. A good example of that would be the cells in our trachea, in the inner lining of the trachea. And those cells are ciliated cells. They have cilia. And the cilia, they beat, right? They beat upwards. So when we have a mucus in the trachea, and the mucus is helping to trap fibers, pollutants, toxins, now we want to make sure that the mucus move upwards so that we can cough it out, spit it out, get rid of it. What is going to allow the mucus to move upwards? The beating of the cilia, okay? In a type of movement that is called the ciliary escalator movement. Now, another structure on the surface of the cell is what we call the microvilli. You know, if the cilia looks like this, guys, the microvilli is even smaller. Is the small little hair-like structures on the surface of the cell. Now, the microvilli is not made of microtubules, and the function of the microvilli is to increase the surface area. For example, where do we find microvilli? We find microvilli in the inner lining of our small intestines. And the reason why the inner lining of the small intestine has cells with the microvilli is, again, to increase the surface area so that we can absorb more nutrients. Okay, the more surface area that we have in the small intestine, the more nutrients we can then absorb. All right, moving on inside of the cell, and I want to start with this guy right here. This is what we call the mitochondrion. Now, mitochondrion is singular, mitochondria is plural, and the function of the mitochondria is that they are the energy. This is where most, not all, but most of the ATP in the cell, this is where they're going to be made. Okay, very, very important organelle. Next, we have what we call the centrosomes. And we can see a centrosome right there. What is the function of the centrosome, folks? The centrosomes are involved with cell division because the centrosomes make little fibers called mitotic spindles. And those fibers, they're going to grab duplicated DNA, they're going to grab duplicated proteins and organelles, and they're going to partition them, separate them inside of the cell before the cell undergoes cell division. Okay, so mitotic spindles are made by centrosomes. A cell that doesn't have a centrosome cannot divide, such as the neuron. Okay. Next, we have this little vesicle right here that we call the lysosome. What is the function of the lysosome? The lysosome is the recycling center of the cell. Why? Because the lysosome is going to take in DNA, old RNA, even bacteria that found itself inside of the cell, and is going to break them down. So it's going to break down the DNA into nucleic acid, break down RNA into the nucleic acid so that they can be recycled. Now, another organelle inside of the cell that is also involved with recycling is an organelle called proteasome, right here. What is the function of this proteasome? The proteasome, folks, they are involved with the breakdown of proteins. Different from lysosomes, lysosomes break down different things. Proteasomes only break down and recycle proteins. Okay? Next, we have another little vesicle right here that is called the peroxisome, and the detoxifies certain toxins in the body. Among the toxins that the peroxisome neutralizes is a toxin called reactive oxygen species, also called the ROS. Now, this reactive oxygen species, these are chemicals that are produced during the process of metabolizing oxygen gas to synthesize ATP. We all know that we need the O2 oxygen gas to survive. The reason why we need the O2 is so that the mitochondria can make ATP. Without oxygen gas, we don't have enough ATP. Now, in the process of converting oxygen gas to synthesize ATP, these compounds here are made, folks. And the reactive oxygen species are very toxic. They can damage and destroy the DNA of the cell. Now, the reason why they're not going to destroy our DNA is because inside the peroxisome, there is an enzyme called catalase. And catalase breaks down reactive oxygen species. Moving on, let's talk about this big organelle right here surrounding the nucleus, shown here in green with all the little blue spheres attached to it. This is what we call the rough endoplasmic reticulum, or the rough ER. What are these little blue spheres attached to the rough ER? These are ribosomes, okay? Bound ribosomes. Now, what do ribosomes do? Ribosomes make proteins. So the function of the rough ER is to synthesize different types of proteins. Okay? Attached to the rough ER, we're going to find this organelle right here. This is what we call the smooth ER. And it's called smooth because it doesn't have all the ribosomes attached to it, which means that it cannot make proteins. Instead, the smooth ER is going to synthesize lipids, including cholesterol. Next, we have the Golgi apparatus, also called the Golgi complex. Same thing, shown here in yellow, folks. What is the function of the Golgi complex? The Golgi complex is what we call the delivery center of the cell. Why? Well, that is because the Golgi apparatus is going to receive the proteins coming from the rough ER, and then the Golgi apparatus is going to package the little proteins and deliver them either somewhere inside of the cell or send the proteins outside of the cell. So the function of the Golgi apparatus is to deliver proteins. Okay? And finally, here in the center, we can see the nucleus. And the nucleus is divided into different parts. Again, we know that the nucleus is the control center of the cell. Now, what are the different parts of the nucleus? The nucleus is made of an envelope, a membrane called nuclear envelope. This nuclear envelope has a lot of little holes, little pukas on the envelope that we call the nuclear pores. And all the way inside of the nucleus, we're going to find this dark region right here. That is what we call the nucleolus. What is the function of the nucleolus? The nucleolus is where ribosomes are assembled. They are put together. Once they are assembled, they are sent outside of the nucleus through the nuclear pores, and then they go into the cytoplasm of the cell to make proteins. Now, outside of the nucleolus, we can see this lighter region right here. This is what we call the chromatin. What is the chromatin? The chromatin is where we're going to find our DNA wrapped around the little proteins called histone proteins. Okay? Any questions, folks? Anything at all? All right, moving on. Now, let's talk very, very briefly about the cell cycle. And we know that like everything else in our planet, every living being, we have a cycle in our life, right? We go through different phases. Now, a cell, any cell in the body, be it a heart cell, a liver cell, a kidney cell, a cell is spent most of the time in what we call the G0 phase. What is the G0 phase? The G0 phase, folks, is a phase in which the cell is doing cell things. The cell is not dividing. A heart cell is doing heart function. A kidney cell is doing kidney function. A lung cell is doing lung function. Okay? Is doing its regular function. Once in a while, these cells are going to get out of the G0 phase and it's going to enter what we call the interphase. What is this interphase? The interphase is a phase in which the cell will prepare for mitosis. It will prepare for cell division. And the interphase is made of three smaller phases. We have the G1, we have the S, and we have the G2 phase. The G1 and the G2 phases, folks, and the G here stands for growth. This is when the cell is replicating all the organelles, all the proteins inside. The cell is very metabolically active, okay, duplicating everything inside in preparation for cell division. Now, what about the S phase? The S phase, S stands for synthesis. What is going to be synthesized? DNA will be synthesized. The cell is going to make 2 copies of that DNA. Now, at the end of the interphase, when the cell is big and ready, now the cell is going to enter what we call the mitotic phase or mitosis. And within mitosis, we're going to find 4 phases. In order, they are the prophase, we have the anaphase, the metaphase, we have the anaphase, and we have the telophase. These are the 4 phases of mitosis, and we're going to talk a little bit more about them in a minute. Now, what is the end result of mitosis, folks? The end result of mitosis is the formation of 2 identical daughter cells that are clones, that are copies of each other. Okay? Now, let me tell you a little bit about the S phase and the DNA replication process. It is a very complex process that you guys don't really need to know in detail. What I want you to understand is that the first thing that needs to happen in order for DNA to be replicated is that the DNA needs to unwind. We all know that DNA is double helical, right? The cell cannot replicate the DNA if it is in the double helical shape. So the cell first needs to unwind these 2 strands, separate them, and once they are separated, now the DNA replication machinery can start reading the nucleotides on the old strand and adding new nucleotides in the growing new strand. Nucleotide by nucleotide. Okay, what are we going to get at the end? At the end, we're going to get 2 copies of this DNA. So now the cell is ready to enter mitosis. Okay, so let's take a closer look at the mitosis then. And again, mitosis includes 4 different phases: prophase, metaphase, anaphase, and telophase. In the prophase, there are 2 important things that are happening, folks. The first important thing that is happening in the prophase is that the centrosomes right here, which have been duplicated by now, they're going to migrate to the opposite ends of the cell. They're going to move away from each other to the opposite ends of the cell. The second important thing that is happening in the prophase is that the nuclear envelope is gone. So we can see the fragmentation of the nuclear envelope. So now all the duplicated DNA, they're going to be floating around in the cytoplasm of the cell. Next, we have the metaphase, and we cannot miss the metaphase. It's very easy to identify. If you look at a cell under the microscope that is in the metaphase, we can see all the duplicated DNA lining in the middle of the cell, guys, in a region that we call the metaphase plate, the metaphase plate. Okay, that is what's happening in the metaphase. Next, we have the anaphase. And in the anaphase, there are 2 important things happening. The first important thing is that all the DNA that is lining in the middle of the cell in the metaphase plate, they are being grabbed by mitotic spindles that were made by the centrosomes. Now, those mitotic spindles are going to retract. Guys, as they retract, they're going to partition, they're going to separate everything inside of the cell, moving them to the opposite ends of the cell. The second important thing that is happening is that we're going to start the formation of what is called the cleavage furrow. And this cleavage furrow is the beginning of the pinching of the cell in the middle. Okay, it initiates that pinching of the cell smack in the middle of the cell. And finally, we have the telophase. And in the telophase, again, we have 2 important things that are happening. Number 1, we are going to have the reformation of the nuclear envelope. We can see right here the cell once again has a nuclear envelope surrounding the DNA. The second important thing is that we're going to have the finishing of the separation of the cell in a process that is called cytokinesis. So what is cytokinesis? Cytokinesis is the actual separation of the cell into two identical daughter cells. And that happens within the telophase. Any questions about mitosis, folks? All right, moving on. Let's talk about the plasma membrane. And again, we all seen this before, and we all know that our plasma membrane is made of something called a phospholipid bilayer, which means that we have two layers of phospholipids on top of each other, right? Now, what does this phospholipid looks like? The phospholipid has a glycerol head, and this glycerol head here has a phosphate group attached to it, which makes the glycerol head charged. It has a negative charge to it. And because of this charge right here, the glycerol head of the phospholipid is hydrophilic. What does hydrophilic mean, folks? It means water lover. It likes to interact with water. Now, the little fatty acid tails of the phospholipid here, they are neutral. They don't have a charge, and therefore they are hydrophobic. They don't like to interact with water. And this is the reason why the head of the phospholipid is either pointing outside of the cell where there's a lot of water, or pointing inside of the cell where there is a lot of water. And the little fatty acid tails, the hydrophobic fatty acid tails, they are hiding in. Understand what the plasma membrane is made of, let's talk about the transport of things in and out of the cell. And because a cell does not have a mouth, the cell does not have an anus to expel the waste, things can only move in and out of the cell either directly through the phospholipid bilayer or protein. Okay, those are the options, folks. So what we're going to see in the next slides, we're going to see what are the things that can move freely through the phospholipid bilayer directly, and what are the things that need the help of a transmembrane protein to move in and out of the cell. Now, when we talk about the transport of things in and out of the cell, we need to understand diffusion. Diffusion is a mode of transport. First of all, that is the definition of diffusion, okay? Now, diffusion follows two important rules. The first rule of diffusion is that in diffusion, things move from a place of high concentration to a place of lower concentration of whatever is that is moving. Again, always from high to low. The second rule of diffusion is that diffusion does not require any input of energy. Why not? Because diffusion drives itself. What is driving diffusion, folks? The kinetic energy of the atoms. Okay? Every atom has a vibrational energy, what we call the kinetic energy. And this kinetic energy, this vibrational energy of the atoms, this is going to allow diffusion to happen. So we don't need to do anything for diffusion to happen. It happens automatically, spontaneously. All right? Now, there are three different types of diffusions in the context of moving things in and out of the cell. We have what we call the simple diffusion. We have facilitated diffusion, and we have the diffusion of water which has a fancy name. It's called osmosis. But again, it's simply the diffusion of water. All right. Now, let's take a closer look at what's happening here with each one of this diffusion. By the way, folks, the activities we're going to be doing today, the two activities we're going to be doing today, they are related with diffusion. They are related with simple diffusion. They are related with what we call osmotic pressure, which we're going to talk more about it next. Okay, so if you don't understand the diffusion, you will not be able to complete the activities today. So this is very important. Now let's take a closer look at each one of the different diffusions here. Starting with the first one. Excuse me. And we can see right here that whatever this is, these yellow little spheres, they can simply move across the phospholipid bilayer. This is what we call simple diffusion because they simply moving across the membrane. They don't need any help. Okay, now what are the things that can move through simple diffusion? Things that are neutral and things that are small. For example, gases, CO2, O2, nitrogen gas, hydrogen gas can easily move through in and out of the cell through simple diffusion because they are neutral and they are small. Next, we can see here, number 2 and number 3, that whatever this is, they require a transmembrane protein to move. Now, in number 2 right here, we can see that whatever this is quite large. And because it is a large compound, it cannot move through the phospholipid bilayer directly. It needs help from a transmembrane protein. So the transmembrane protein will facilitate the diffusion of whatever this is. Okay, so large molecules like glucose, like proteins, will only move using a transmembrane protein. What about number 3 here, folks? Here we can see whatever this is looks quite small. So the question then is, why is not moving through simple diffusion? Why does it need a transmembrane protein? The answer is because it is a charged molecule. Charged molecules like sodium ion, potassium ion, calcium ion, magnesium ion cannot move through the phospholipid bilayer because they are charged. And the fatty acid tails right here, which are neutral, will repel anything that is charged, with the exception of water. Because as we can see right here, number 4, water can move any way that it wants. It can move through simple diffusion, although it's not very efficient, but it will mainly move through facilitated diffusion with the help of a transmembrane protein called aquaporin. Okay, and again, keeping in mind that the diffusion of water is what we call osmosis. It doesn't matter if it is simple diffusion or facilitated diffusion of water. If it is water diffusing, we call it osmosis. Any questions, guys, about diffusion? Keeping in mind, however, that we can only use diffusion when it's moving from a place of higher concentration to a place of lower concentration. What we call moving down the concentration gradient, okay? From high to low. Now, let me talk a little bit about osmotic pressure, because again, it's something that you guys will do today with the in-lab activity. And I want to explain to you what osmotic pressure means, okay? It's not complicated at all. And to explain, to talk about osmotic pressure, folks, I want to use a device and do a little experiment right here. And this device is what we call a U-tube. Yes, before the U-tube, there was a U-tube, a tube in the shape of a U, okay? And in this U-tube here, In the middle of the tube, we have a selectively permeable membrane. What does that mean, selective? It's selective only for the movement of water. It does not let anything else but water to move across. Okay? Now, on the left arm of the U-tube, we have 100% water. Just H2O, nothing else. On the right arm of the U-tube, we have 50% water and 50% solutes. Whatever the solute might be, salt, sugar, it doesn't matter. All right. Now, when I let the system run, folks, where will the water move mostly? The water will move mostly, as we can see right here, the water will move from left to right. Now, the question is, why is the water moving mostly from left to right? And the answer is because water is moving down the concentration gradient from 100% on the left arm to 50% in the right arm. Until when, folks? Until the system has reached equilibrium. What does equilibrium look like? In equilibrium means that both sides of the membrane will have the same concentration of water, both on the left arm as well as in the right arm. Now, you might be asking yourself, well, wait a second. This doesn't look in equilibrium. It seems like there's more water on the right arm than the left. And that is not true, folks. You have to account for the fact that on the right arm, there is a solute, which is going to increase the volume. Okay? But the amount of water, the concentration of water on both sides of the membrane is the same. The system has reached equilibrium. Now, what if I want to bring the system back to the starting condition? What do I need to do? Folks, what I need to do to bring it back to what it was before, I needed to apply pressure and push the water to the left arm right here, where the water does not want to go. Now, the amount of pressure, the amount of force that I use to push the water to the other side of the membrane, this is what we call osmotic pressure. Whatever that the pressure might be, that's what we call osmotic pressure. Let me give you a real life application of osmotic pressure, okay? Most of the water that we drink, the bottled water that we drink, that we buy at the supermarket, that water is purified by a process called reverse osmosis. What is reverse osmosis, folks? Reverse osmosis is exactly what we just did right here. It should take a water, let's say you take water from a river, okay? And you're going to take the water and you're going to pass it through a membrane. You're going to force the water through a membrane, and that membrane only allows water to move across, nothing else. And on the other side, what do we get? We get pure potable water that we can drink. And that potable water, that clear water, clean water, it was produced by reverse osmosis. Okay. All right, moving on. Let me talk now a little bit about tonicity, which is something that you guys also will see on the lecture exam tomorrow. And tonicity is related with the movement of water. It is related with osmosis. However, tonicity explains the amount of solute dissolved in a solution. Again, it is water that is moving across the membrane, and the membrane is the cell membrane, the plasma membrane. But tonicity is meaning, the meaning of tonicity is the amount of solids in the solution. And a solution can be either isotonic, hypertonic, or hypotonic. An isotonic solution, it means that the amount of solutes that we have in the solution is the same as the amount of solutes inside of the cell. Another way to look at this is that we have 95% water in the solution and 95% water inside of the cell. The system is in equilibrium. Where will the water go, folks? The water will move equally in and out of the cell. Okay, the amount of water going in the cell is the same as the amount of water going out of the cell. The system is in homeostasis. And this is the ideal solution for a cell to survive. Our blood is an isotonic environment, an isotonic solution. A hypertonic solution, this means that the amount of solutes in the solution is greater than the amount of solutes inside of the cell, which also means that we have 80% water in this example here, 80% water in the solution and 95% water inside of the cell. Where will the water move? It will move down the concentration gradient from 95% inside of the cell to 80% outside of the cell. What's going to happen to the little cell, folks? The cell is going to shrivel, and die by a mode of cell death that is called crenation. So what is crenation? Crenation is when a cell loses water, shrivel, and die. Now, what about a hypotonic solution? In a hypotonic solution, we have less solutes in the solution than we have inside of the cell. Now, let's look at the water. So in this example here, we have 99% water in the solution, 95% water inside of the cell. Where will the water mostly go? Down the concentration gradient, from 99 outside of the cell to 95 inside of the cell. Now, what's going to happen to the cell here, folks, in a hypotonic solution? The cell is going to take on water and it will literally explode. And it's going to die by a mode of cell death that is called lysis. Okay? If it shrivels and dies, it's called crenation. If it takes on water and explodes, it's called lysis. Any questions? All right. And the last thing I want to mention is that so far, when we talked about diffusion, folks, diffusion is a passive process. It's a passive transportation. Why is it passive? It is passive because it does not require any energy. Now, the caveat here is that for diffusion to happen, it can only happen if things are moving from high to low concentration. Now, sometimes the cell needs to move things against the concentration gradient from low to high. Now, when we want to move things from low to high, we will need to spend energy in the form of ATP. And that type of process is called active process. Okay? So active process requires ATP and is used when we want to move things from low to high against the concentration gradient. All right, folks. So let's talk about the in-lab activity for today. So as I mentioned, we have two different activities. Activity one, which is simple diffusion, and activity two called osmotic pressure. So we're going to simulate simple diffusion. We're going to simulate osmotic pressure. Where do we find the activities? How we do them? So let me share my screen here with you for a second. So if you go to our Lamaku site, right there, and if you click on content, right, you go to content, and then you go to week 4. We're already on week 4. Click on week 4. You're going to find all these different folders here. So you have the lab manual, in-lab activity, in-lab quiz. So click on the in-lab activity folder. A document is going to open up right there. So guys, here you're going to find the 2 links for the different activities. You're going to have a link for the simple diffusion, and you're going to have a link for the osmotic pressure. Both of them is going to take you to the same website, different activities. Okay, there are 2 activities to complete. Let's click on the first one here and do the beginning of them together. So when you click on that, this website is going to open up. So you have the PEX, Physio-EX simulating dialysis, simple diffusion. Please read through the objectives here real quick. You can also just browse through the introduction. There's going to be a little pre-lab quiz before you get started with the experiment. I need everybody's attention here, please. These are not our in-lab quiz. Okay, these are separate things. The in-lab quiz, you're going to do it after you're done with the in-lab activities. This pre-lab quiz here is just to see what you know before you start the activity. So they're not going to be graded, but I do want you guys to answer them. Answer them and click next. Then you're going to find this note here, click submit. And now you are at the first page of the experiment. Guys, the experiments are very simple, very straightforward. And it's going to give you clear instructions on what you need to do step by step. So let's do a few of them here together, okay? So you understand what you need to do here. All right, so first here it says, drag the 20 MWCO membrane to the membrane holder between the beakers. Now, what is an MWCO? MWCO stands for molecular weight cutoff. Molecular weight cutoff. Where do we find the membranes? Folks, if you come right here, on the right-hand side, it says dialysis membranes. There are 4 of them. A 20 MWCO, 50, 100, and 200. What does it mean, these different membranes with different numbers? Guys, the numbers on the membranes correspond to the size of the holes on the membranes. If you have a 20 MWCO, imagine the hole is a tiny little hole through the membrane. If you have a 200, you're going to have a big hole through the membrane. So the smaller the number, the smaller the hole. The bigger the number, the bigger the hole. Okay? So it says right here, drag the 20 MWCO between the beakers. So you're going to hover over it, click on it, and drag it in between the beakers. So we did step number 1. Click next. Now it says, increase the sodium chloride concentration to be dispensed to the left beaker to 9 millimolars by selecting the plus button beside the sodium chloride display. Select dispense to fill the left beaker with 9 millimolar sodium chloride solution. So if you come down here, guys, on the left-hand side, we can see all these different solutions. We have a sodium chloride, urea, albumin, glucose. The instruction said, select, add 9 millimolars of sodium chloride to the left beaker. So you're going to come here on the sodium chloride, click the plus button until you get to 9, and then you're going to click dispense. We just filled the left beaker with 9 millimolar sodium chloride. Click next. So now it says, note that the concentration of the sodium chloride in the left beaker is displayed on the left window. Select the ionized water and then select dispense to fill the right beaker with the ionized water. If you scroll down here, you have the ionized water. Click dispense. So now we fill the right beaker with the ionized water. What is the ionized water, by the way? It is pure water, just H2O. There is nothing dissolved in the water. just hydrogen and oxygen. Okay, click next. So now it says, after you start the run, the barrier between the beakers will descend, allowing the solution in each beaker to have access to the dialysis membrane. You'll be able to determine the amount of solute that passes through the membrane by observing the concentration displayed to the side of each beaker right here. A level above zero in the sodium chloride concentration in the right beaker indicates the sodium and chlorine ions are diffusing from left beaker to right beaker, passing through the selectively permeable membrane. Note that the timer is set to 60 minutes, which the simulation compresses to 10 seconds of real time. Select start to start the run and watch the concentration display to the side of the beaker. So scroll down here, click start. We can see the timer here going down to zero. Wait until it goes down to zero. All right, now it has a note right there. It says no diffusion of sodium chloride. I have a question for you. Why not? Why didn't the sodium chloride diffuse from the left beaker to the right beaker, folks? What is the reason? Don't overthink it. Don't overthink it. Remember, the membranes have different size of holes, different
The pore
pores.
size was too small?
The pore size was too small. Absolutely. That's the reason why the sodium chloride didn't go across, because it couldn't fit through the 20 MWCO membrane. Okay? And these are the type of experiments you're going to be doing. You're going to be testing the different membranes and the different solutions. All right, guys, let me move forward here. So now, when you get to the end of the experiment, it's going to say right there, and when you get to the end, it's going to say, congratulations, you have completed the experiment. Select submit to record your results in the lab report and proceed to the post-lab quiz. You're going to click submit. Submit again. And now you're going to have a little post-lab quiz. Again, this is not our in-lab quiz. Please answer them. They're not going to be graded, but I do want you to answer them. And click next. Click submit. And now you're going to get to a review sheet. Everybody attention here, please. Stop doing whatever you're doing. You don't need to answer the review sheet. Once again, you can ignore the review sheet, okay? Just leave it blank and click next. Submit. And there you have it. You have your lab report. If you scroll down all the way to the bottom of the lab report, you're going to see here this button. It says printable, savable version. Click on it. Enter your name. And now you're going to have an option to print or save as. Click on that button. Now, of course, you don't need to print this out. I want you to save this as a PDF file. Make sure you know where you're saving it in your desktop so you can find it. Okay? You're going to save it as a PDF file, and then you are going to email those 2 PDF files to me. I need to have them by the end of class. Okay? Now, some people in other classes told me that they were having trouble saving as a PDF file. If you cannot save as a PDF file, folks, when you get to the end of your lab report, take screenshots of the lab report, attach the picture files to an email, and send them to me. Okay? A PDF or a screenshots of the lab report, it doesn't matter. I accept both of them. But I need to receive them by the end of class today. Otherwise, you're not going to get points for the in-lab activity. After you send the email with the lab reports to me, Then you're going to go back to Lamaku and you're going to take the in-lab quiz for today. And after you're done with the quiz, you're good to go. Guys, any questions? Do we know what we need to do? Awesome. Let's get started.